NanoGrid technology utilizes array-capture DNA and gold nanoparticles to detect targets. Each nanoparticle is functionalized with universal oligonucleotides that will specifically bind through a mediator to the target nucleic acid that is hybridized to the array. The gold nanoparticles are 13 to 20 nanometers in diameter. 1 nanometer is approximately 1/10,000th of the width of a human hair. NanoGrid technology utilizes slides covered in spots. These spots contain strands of DNA complementary to various targets of interest. The type and the layout of the DNA spots will vary depending on the assay. Each spot on the grid contains DNA for a specific target or control nucleic acid. Sample nucleic acid that has been extracted and/or amplified for the targets of interest washes over the slide containing the NanoGrid, which is housed within a hybridization chamber. The temperature within the chamber increases, causing the sample nucleic acid strands to denature. Then, the chamber is cooled, and as the temperature within the chamber drops, the denatured strands attach to complementary target strands on the NanoGrid, if present. The remaining unattached DNA strands are flushed from the chamber. Next, a mediator sequence of DNA and gold nanoparticles are introduced into the hybridization chamber. One end of the mediator strand is complementary to the end of the target nucleic acid that is not bound to the immobilized spot. The other end of the mediator is the poly A tail, which does not bind to any sample nucleic acid. The gold nanoparticles are covered in a poly T tail, the complement to the poly A tail on the mediator DNA. This interaction allows the mediator DNA to bind to the gold nanoparticles. The remaining unattached mediators and gold nanoparticles are flushed from the chamber. Finally, a silver solution is washed over the slide. The silver sticks to the gold, continuing to build on the gold and makes the particle larger and larger. LED lights shine across the surface of the slide, and a camera within the instrument takes an image of the slide at multiple exposures. The silver coating scatters light back to the camera, creating a visible spot on the slide if the target nucleic acid was present in the sample. The spots with no complementary sample nucleic acid attachments, and therefore no silver, do not scatter light back to the camera and remain dark. The instrument analyzes the slide to determine the presence or absence of light scatter. NanoGrid technology provides an effective multiplexing solution. Multiple targets are arrayed on one slide. Controls are placed at each end of the slide, setting the grid and allowing the instrument to analyze data quickly and accurately. NanoGrid technology allows increased sensitivity by several orders of magnitude compared to fluorophores. High specificity is enabled for both nucleic acid and protein detection. The gold nanoparticles are extremely stable, have a long shelf life, and are non-toxic.